Oh, thank you. Is it on? Oh, it's not. Okay. Cool. All right. I think we'll get started now, if everyone's ready. Cool. All right. Thank you so much for coming to the inaugural BioML seminar to kick off the spring semester. Really excited for the lineup we have this semester. We're going to have people like Sam Rodrigues. We're going to have Tom Kalo. We're going to have Riju Das from Stanford and a couple other people in the works. So yeah, to introduce our speaker, our first speaker of the semester is Sam Rodrigues, who is CEO of Future House, a non-academic research institute developing semi-autonomous AI in service of scientific research. Future House's recent work includes WikiCrow, which is a retrieval augmented generation agent for querying and summarizing scientific literature, and BioPlanner, which is an automated framework for evaluating scientific protocols. Apart from his work at Future House, Sam is the founder and PI of the Applied Biotech Lab, an academic research group at the Francis Crick Institute in London. Some of Sam's notable inventions include a new nanofabrication method, and a technique for neural activity sensing. We have bloodstream probes, and a method for protein sequencing, and a technique for mapping brain connections. So give a round of applause for Sam Rodrigues.
Thank you.
Attach this to your,
Oh, yes.
yeah,
Good.
and then send this to your shirt if you want.
Great.
Here you go.
Is this good?
I'm gonna just leave that because I don't know how to bring that thing back after I put it away.
Awesome. Thanks.
Thanks, guys. So first of all, thanks for having me. Super excited to tell you guys about what we're doing at Future House. I thought I would editorialize for a little bit at the beginning on 2 points. The first point is I heard that this seminar is funded by or sponsored by Pillar. Tony Kalesa is a really good friend of mine who is I don't know if he's a partner. If he's not a partner yet, he'll be a partner very soon, I think. Anyway, the point is he's an investor at Pillar. And I think the thing, he's also like a founding member of the board of directors at Future House. I just something to say on that is that like for people who are thinking of starting their own companies, when I was in your position, the way I would think about it is like, oh wow, like if you have an idea for a company, you have to go out and you have to find some investors who like really want to invest in you. Well, it turns out that like the best companies get to choose which investors invest in them, right? Because they're like the top 1% are the companies that all the investors want to be piled onto, and then you get to pick and choose. And when you're thinking of picking and choosing, you should be picking on the basis of like which of those investors are going to add value, because it turns out like everyone has money. Money is like a pretty abundant commodity. But like some investors will let you take it to the next game, or to take the game to like the next level. And like Tony is a VC that you should take money from. Like if I had to take money from like any one VC, it would be Tony. Okay, like he's just like, and as you guys get to know him, I hope you all get to know him. Like the value add that he provides for people who are starting new anything is just like insane. Remember, Future House is a nonprofit, right? And still, like Tony's just like available for a call like whenever you want. Like I guarantee you, I could like pick up the phone right now and call him and like ask him to help with something, and he would like be there, right? And he makes no money. So okay, so that's Tony. He's awesome. Strongly recommend. The second thing is I'm going to tell you guys about Future House. Future House is a like philanthropically funded like nonprofit research organization. And I'm going to tell you about our mission and everything. I wanted to say a little bit just like right at the very beginning about like this idea of professional research environments. So like also when I was sitting, in your guys' position, and I was like going through my PhD, I was inspired to tell this story by a conversation that was happening over here about people deciding whether they should go to grad school or not, right? Like I, like many of you, probably like really wanted to be doing research. Or I should say, like when I was in grad school, I like still like really was looking forward, after I was done with grad school, and like really wanted to be doing research, and thought it was insane that like certainly in bioengineering, there was basically like nowhere where you could go where your job was going to be to do research, except in a university. And if you're in a university, you had to be a trainee, right? So like, you're like undergrads are obviously, students. Graduate students are still basically students, get treated like students, and even postdocs, they're like called postdoctoral trainees, right? Like, the point is that like, the idea is that this is like an itinerant position. It's not like your career is to do research, you just get to sit there. And then one day you become a professor, which means finally your job is to do research. And what do you do all day? You sit in your office and you write grants, right? So I just thought that was insane. And it like led me to really wish like, hey, shouldn't there be a place where you can go where like professionally you'll be able to do research? And that led to this idea of these focused research organizations, which is this idea that Adam Marblestone and I came up with towards the end of my PhD, which are basically like these philanthropically funded nonprofit startups for science, for projects that like don't fit in academia because they require a team that's larger than what you can do in academia, but also can't be done effectively for profit. And like they actually provide a pathway for people to like kind of do research like as a career professionally. And the way that they will become like, so there are several FROs now. Most of them are funded through Convergent Research, which is something that Adam Marblestone runs. FutureS is not part of Convergent, but is also basically an FRO. And what needs to happen in order for them to become like a mainstream part of the scientific research ecosystem is for the government to start funding them, which it will do once we have like really good proofs of concept that the system works. And I just really hope that happens, because I'm a really strong advocate for that happening, because I think that like in biology, certainly there are very few, maybe no places where you can go and like be an individual contributor and like do research professionally. In machine learning, there have been. And now those places still are like becoming more commercial because the commercial applications are being realized. And so I think that this is an important part of the ecosystem that's missing. The ironic part is that that was what I wanted, to find a place where my job could be to do research. And I created such a place. And now what do? I sit in my office and I don't write grants, but I like run the organization. And so it's, such is the life as a founder. Good. So basically now FutureS. So thank you guys for coming to listen. So let me see if I can make this go. Great. The past 10 years have seen like really incredible advances in the tooling that we have for biology research. So maybe to put a little bit more context on this, I was a theoretical physicist originally. I did my early research on like quantum information theory. And I got into biology because it seemed like there were going to be more unsolved problems there. And I got into biology around like 2013. And it was like immediately apparent that biology was like very strongly data limited. Like we just did not have the tools that we needed in order to like ask any of the questions that we wanted to ask. But in the past 10 years, there's been this explosion in like the different kinds of tools that allow us to gather data and test hypotheses in biology. So things like massively parallel oligonucleotide synthesis, which allows you to basically like, well, as it says, synthesize hundreds of thousands of oligos at a time, where each oligo can represent a different kind of experiment. It can represent a different protein that you want to test, a different enhancer or whatever, right? Droplet-based single cell sequencing, so that you can like now sequence actually like hundreds of thousands of cells at a time, where again, each cell can be a different experiment because each cell can like encode a different protein. Obviously next generation sequencing. And so the result of all of these things is that like we actually are now like strongly bottlenecked in many areas by like human bandwidth in interpreting the data that we get, right? So I think that the best example of this is that the GTEx consortium, this was one of my favorite ones, back in March, like last March. The GTEx consortium, which is a big consortium at Broad, released their new data set and published it as 15 papers in Science. The Allen Institute, it's not just the Allen Institute, there's a consortium, like Brain Cell Atlas Consortium in December, that when they released their data set, they published like 10 papers in Nature. And okay, so the point at which you release a data set and your initial data dump is like 10 papers in Nature, you know that there's like, you're like limited now by literally your ability to just like go and get those insights and publish those papers. Like there's a lot more data in those data sets that we're not, a lot more insight to be gained from those data sets that we're not extracting. And so our mission at Future House is basically to build an AI scientist in order to scale up discovery research in biology. And so the important thing here is like discovery research. So we're not, like the goal is like not to make a drug, or like maybe the long-term goal is to make a drug, right? Or to make drugs, like make lots of drugs. But like, you know, the point is that my thesis at the moment is that we can't make drugs for the diseases because we don't know how the diseases work. And so it's like garbage in, garbage out. If you have like a new way to like make a better molecule, but you're going after the wrong target, your chance of success is going to be like exactly the same as everyone else's. We want to understand actually how biology works. We want to be creating mechanistic models. What's up? Yes.
That we're recording because they wanted me to double check. Are we recording?
Yep, you're recording.
Okay, good.
There you go.
Perfect. All right, show goes on.
Awesome, great. So thank you. Okay, and so the way that we're going about this is basically thinking about how do we automate, like, how do we build automated systems that are going to be able to extract insights from these big data sets that will be able to figure out where the gaps are, come up with like new experiments that they want to run, and ultimately figure out how to interpret those experiments also. And so these are some of the areas that we're thinking about, like literature search, kind of hypothesis generation, analysis, basically like as a way of getting a foothold on the problem, thinking about what are the various steps of the scientific process that we go through, and then how do we figure out how to automate and parallelize them. And so we have this like challenge book that kind of describes the set of problems that we would like to be able to solve once with an AI scientist. And examples are right like things like the examples are generally problems that are kind of like too big for humans in some way, right? So like generating a comprehensive network of all known or proposed like protein-protein interactions for the human proteome, like, figuring out how to go and like create like a table that shows you all the DNA binding sites for transcription factors in the human genome, right? These kinds of problems are generally of the form like for all X do Y, where Y is something that a human can do, and when you add but like adding the qualifier for all X makes it both like way more impactful and like completely intractable, right? So like if you want to understand like the protein interactions for a single protein, that's like pretty doable. If you want to go do it for the whole proteome, then it becomes very hard. And so why is this interesting? This is interesting for two reasons. The first one is that actually like most of biology is too big for humans, right? So there are 20,000 genes in the genome, there are tens of thousands of different cell types. I have no idea how many metabolites or small molecules there are. Then there are like, hundreds of millions or more organisms. I have like a kind of fringe hypothesis that like, because I did my PhD in Ed Boyden's lab, and anyone who has met Ed, like his whole thing is we have to solve the brain, right? Like, I have a fringe hypothesis that the brain is actually already solved, but that like we don't know it because like no one has the time to go and like get through assemble the model from like what is already known in the literature, right? Like that's not not exactly serious, but like the point is that if like the information did exist in the literature to like solve the brain, whatever that means, we would not know. Right? Like no one has the time to go and like read all the literature, process all that information, come up with a model. Okay, so that's reason number one. Reason number two is that actually I think like science is like the perfect playground to develop AGI, whatever you want AGI to mean for you. It's maximally open-ended, right? It uses like most of the higher order cognitive functions that humans have, like lots of like, and kind of in their most extreme form, right? Like, motivational, like the kind of motivational reasoning, or like how figuring out like how do humans go about creating the reward functions that we pursue. Like becomes a really interesting question in the absence of like any kind of objective reward. It becomes a really interesting question in the context of science, because it's like you start your PhD and you have like some hypothesis about like what will get you what counts as reward. Maybe you're like, oh, what I want is like a Nature paper, and then you have some hypothesis about like how to go about writing that Nature paper. You don't really know, and like you're integrating information, like super sparse information signals, super sparse information to kind of refine that reward function. It gets it ends up being this environment, we think, where you can you get a lot of these cognitive functions in kind of their most extreme form. It's pretty interesting. It reveals a lot of interesting problems that seem like they're on their way to AGI. So interesting for those reasons. So key facts about Future House. This line at the top got added in the 3 minutes before the talk began by, inspired by the conversation. We do research and we pay money, which is a differentiator. So we're an independent nonprofit. We're based in San Francisco in the Dog Patch, funded by Eric Schmidt, although that's no longer exclusively funded by Eric Schmidt. We now have other funders, although we'll make like an announcement about that at some point. But Eric was our like main backer. We got started in August, or we got started in September. We announced in November, and I think that one of the cool things is that we like have a wet lab also, and so one of the inspirations behind going to Future House was wanting to have a place where you have AI researchers, like AI engineers and like wet lab biologists all in the same place, so that you get this really tight feedback loop. And that was really essential. I'll tell you guys about WikiCrow. That was really essential in WikiCrow. And like it's at the top, I mean, like we're focused on actually like inventing new techniques and like coming up with new fundamental breakthroughs, but we pay industry competitive salaries. So and we're currently 8 people, but we'll be 11 on Monday. Is today Monday? No. Yes, today is Monday. But a week from today, we'll be 11. So we're growing fast. Okay, and this is the leadership team. So I'm the director and CEO. Actually, I am now everyone at Future House. So Future House is a super flat organization, and so everyone is either a member of technical staff or the head of something. And it was very awkward for a while that my title was director and CEO. So it just got revised. I'm now head of Future House. And then Andrew White was a professor at the University of Rochester, is now head of science. Matt Rubashkin has built kind of like scalable ML systems at a bunch of different companies, now head of engineering. And we may be expanding our leadership team as well. So these are our 3 focus areas, and I'm going to tell you now about the literature search topic. This was like our first technical result that we came out with, which was WikiCrow, which was in early December. And the idea here is basically, like I was saying before, I mean, how do we go about automating synthesis of human scientific knowledge, right? I mean, the scientific knowledge is like extremely dispersed, and the literature is way too vast for any one person to understand. This problem became like very obviously much more tractable with like the dramatic improvements that we have seen in language models. So if now is obviously the right time to do it. So WikiCrow, in order to tell you about WikiCrow, we're going to start with paper QA. So paper QA is like, it's basically the algorithm that underlies WikiCrow. And the basic idea behind paper QA is to be able to take a very technical question, and like this question on the left about RAS isoforms, and be able to provide a cited, technical and accurate answer. And I'm just gonna, on that point about accurate, because I'm gonna say this a few times, but if there's any one thing that you guys should take away from this, it is that like 90% of the problem is being able to measure performance. When we're like working on these problems, like building an AI scientist involves like these like very nebulous like things like hypothesis generation and blah blah, and like you can't get any traction unless you can measure performance. And so just spoiler alert, the thing about WikiCrow that is cool is that we actually get like a real world objective measure of the performance of these systems, which is the only like kind of real world objective measure of the performance of systems like this that I have seen. So yes, so anyway, the goal is to be able to return an accurate summary of the result, the answer to this question. And so this is the general flow. So this is a RAG agent. RAG is retrieval augmented generation. So pretty big topic now, basically just to be able to find, in order to answer a question, be able to go and find relevant resources and return them, and so and then answer a question that's informed by them. And so the basic flow is that you have a question, like this question here about double Corton. You go and you find relevant papers. You then find chunks, relevant chunks within the paper. And you basically you like chunk the paper and you ask for each chunk whether the chunk is relevant. And then once you have the relevant chunks, you go and you generate generate like a final context by combining the relevant chunks, and then you provide an answer. Now, the thing that is cool about paper QA is that this is paper QA is an agent, which means that at every point it gets to look and see whether it feels like it has enough evidence to answer the question, and if it does not have enough evidence to answer the question, then it can go and retry. And that's kind of what is so it has like the agent has access to these three tools, which is that it can search for papers, it can gather evidence, and then it can finally try to answer the question. Gather evidence means look at the papers and look at the chunk the papers and look at which chunks are relevant. So for example, sometimes it does a search and it turns out that the papers that it comes up with some papers, but it's only able to access the PDFs for some fraction of them. So then it might search again, and then it can gather the evidence. It can look and decide whether it has enough evidence, and so on. And so the first thing that we did after we built this was that we wanted to go and measure its performance on like the standard benchmarks, right? And so the first benchmark that everyone uses is the US medical licensing exam. It's on par with PB4. I mean, this is like not super. Basically, it's like not super interesting. The US medical licensing exam, it's just like very much textbook knowledge. The more interesting one is like PubMed QA. So PubMed QA is a benchmark that is basically built by answering questions that are contained in the abstracts of papers. And the standard version of the benchmark has like the context in the question. So you like provide it with the abstract or a snippet of the abstract. And so we did like a closed book version of PubMed QA, which is basically you just like don't provide it with the snippet. And then obviously, like now it would be very surprising if this were not the case. Paper QA does much better because what it can do is it can go and find the relevant paper, and then it can get the answer. These like are slightly these were like still like slightly unsatisfying as benchmarks. Oh, this is just, I mean, this is more funny than anything else. I think before I tell you about the unsatisfactoriness of those benchmarks, one of the cool things about RAG agents is that they like the RAG like really suppresses hallucination. So this graph got us in a lot of trouble with the reviewers because like we over 260 or 237 like evaluations, we did not like observe any hallucinations. It's just like very well grounded, and this took some prompt engineering also, but it was like very, very well grounded in like in the sources, which is great. But then the problem is like no one believes it, so so anyway, issues with the benchmarks. The first one is like there's definitely a huge contamination issue. The benchmarks are old. They are only from abstracts. Abstracts are like very easy to parse compared to full text papers. They don't require any kind of synthesis, right? And in more general, it's like it's a synthetic task. And so it doesn't like, these benchmarks, like they don't reflect kind of like any real world kind of thing that you want to do. And so we'll talk about the first three problems, and then we'll talk the synthetic task part we'll talk about later. But so the first thing that we had to do was we had to come up with a more challenging benchmark, one that would be better able to distinguish between the performance of like a RAG agent and the performance of like a base kind of model. And so that meant that we need a task that could like provably like require retrieval, right? Which means that we wanted to have questions from papers that were basically like after the cutoff of the models where you were pretty sure that the information in the paper was not findable anywhere except for in that paper. Or ideally in a couple, in like maybe you had to synthesize it across a couple of papers. This is really hard. Like these papers are really, like these questions are really hard to generate. Because proving to yourself that there's a fact that is like actually novel to that paper that has not been reported before. People don't tend to tell you when they publish something that has actually been reported before, like they don't broadcast that. And so this becomes like really, really challenging. But so these are just some examples of what these questions look like. Has anyone performed a base editing screen against splice sites in CD33 before? You can find the paper where people have, and you're like pretty sure that, and then you convince yourself that that is in fact the first time anyone has done it. And you can obviously come up with a lot of questions like that where you change CD33 to CD32, and no one has done that, and so you can generate questions that way. Then you get to ones that are kind of like bizarrely specific, like this one about glycoRNAs, which glycoRNA does not show an increase in ML macrophages in mod simulation, blah blah. So you find a paper about like glycoRNAs in macrophages and et cetera. So this is the kind of benchmark. And so the attributes of this benchmark, there are 50 questions. They're multiple choice. They require full text search. They are after GPT-4 Claude 2 cutoffs, which also means that this benchmark, if you're going to use it, needs constant upkeep. Sucks, but I don't have a better solution to that at the moment, right? Because the cutoffs keep moving up. And it's focused on biology. And this is just like manually curated. Why 50? 50 is really small. While this was kind of for, this was mostly, this gets to the point about like wanting real world tasks, which we'll talk about later. So like mostly for internal use. So we haven't scaled it. We didn't scale it up previously. We are now scaling it up. But, and so this is the performance. We were pretty excited about this. We got humans to do it. And when the humans do it, you just like give them a set amount of time. Right, and you let them, and we chose the amount of time arbitrarily. I think it was like 5 minutes or something like that. So random accuracy is 25.7. The humans are 87.9, and paper QA is also 87.9. So that is, so that was pretty exciting. This is like human level recall for these questions. Something to point out here: Perplexity, the Copilot version of Perplexity. For people who don't use it, the Copilot version of Perplexity is very, very good. I actually like there are several of these kind of commercial systems. None of them release performance metrics, but like Elicit, Cite, Perplexity is like clearly the best. And anyone who has tried using Perplexity, it's like becoming obvious that Perplexity is like pulling away at this kind of a task. So really strongly recommend it. It's great. We use Perplexity internally a bunch. Yeah. So this is not that interesting. Okay, so I keep going on about this real world task. So this is what it was. So after we came up with the system, we're like, okay, what can we actually do with this that will be interesting? And so this is WikiCrow. Previously, at the beginning, so there are 20,000 genes in the human genome. At the beginning of December, there were Wikipedia pages for 3,639 of them. And so we said, okay, like, we've done all this engineering work to create a system that we can like run at scale. So let's go and just write the Wikipedia articles for the remaining 15,600. And then we were like, this will be like our real world evaluation task. Cause like, this is a task where that like, you're not going to be worried about like, contamination. You can compare it if you want against like this is like the task that this is like the performance that you care about, right? Like, it's not, you can't like hack it. Right? This is just like, if you can write the Wikipedia articles, if they're good, if they're comprehensive, if they're accurate, then you win. Because like, this is like an example of the real thing that you want these systems to be doing. Incidentally, there are still 3,639 articles on Wikipedia, because not for the reason that you might think, which is to say that we did write the articles and Wikipedia has not adopted them. And the reason is not because they're written by AI. Actually, Wikipedia is super into AI generated content. Wikipedia is a fully volunteer-run organization, and we went to them and we were like, we have 15,000 articles to add to your corpus. And they were like, we have 2 people who maintain our molecular biology articles. They were like, we cannot accept these articles. So I would love to find a creative solution to that, but that's just that's how it is at the moment. So they're hosted on our website instead. But so basically, how Wikipedia, you know, what we did was that we took a Wikipedia article, and thankfully, like Wikipedia had a spec, like they have a spec on online for what you should, for what a Wikipedia article should contain. And so you just take that and you kind of like turn it into a prompt. I mean, you do some prompt engineering, obviously, but you basically just take it and you turn it into a prompt. And this is just an agent, so you can just give it the prompt, right? And then the agent will figure out that it has to go and run itself several times. It's pretty cool. And so what this boiled down to was executing paper QA 100,000 times in 2 days. We drew from 871,000 papers. Our corpus of full-text papers is 80 million papers. So this is like, these articles are drawn from like 1% of the entire like scientific literature, which is pretty wild, actually. 300,000 searches. And the really cool thing is that there are 670 articles that like very persistently failed. That is to say, there are 670 articles that paper QA cannot find any information for the gene. Okay, so like there are still 670 genes that are just like, they do not appear in the scientific literature. They usually have like these kind of like procedurally generated names, right? This is like, you know, C3 or 15 or something. It's like chromosome, it's like the 15th ORF on chromosome 3 or something. Anyway, so they're annotations, but they don't have any information about them. So examples, I guess I can just can I do this? I think I can do this, right? I can just do is this going to mess up the Zoom? I hope not. Great, perfect. So these are just to show you guys what this actually looks like. You know, we can come through and do literally any, like these are genes. These are all gene names, right? So it's like a little bit alphabet soup. Sorry, and you probably can't read it, but you know, stuff like MIEF2. The one I'm looking at is MKKS. I've never heard of these. These are like 100 randomly displayed ones, but like you can go in and you can find you know, any gene that you want. You can go in and you can look, you know, and you just get this really nice article, like well cited. I don't know, it's a centrosomal shuttling protein. I'm not going to spend the time to read it right now, but you get a structure section, a function section, and it's all cited like this. Okay, I'm just going to go back because I want to make sure that we have enough time to talk about the interesting things. Maybe for people, I don't actually know like what the background is here, like what fraction is AI and what fraction is biology. So maybe just a quick section, quick side note about why you want this. If you do research in genomics, like your life consists of like running some assay and coming back with a list. The assay comes back with a list of like 50 or 100 genes and that are like significant according to that assay, right? Like maybe these are genes that like turn on in whatever biological process you're studying. You have never heard of any of them, right? Because there are 20,000 and the lists are usually basically random. And so you take the first one, you go to Wikipedia or you go to Google and you type it in and up comes nothing, right? Because like the vast majority of genes, just like 80% of the time, I mean, like most genes just like don't have like plain text annotations. And so you have to go back to the primary literature. It takes forever. It's impossible. And so this is like the actual use case here is that it just makes it way easier for biologists to get information out of like out about genes. Okay, so this is now the really interesting thing, which I've been making this whole fuss about real world evaluation. So here it is. The way that we evaluated it was that we took 100 statements and we from each of like, you know, natural from like human generated Wikipedia and from Wikicrow, and we asked for every statement, is it cited and is it like accurate as cited, right? So like, which is to say, like, is it accurate as per the citation? Does it agree with whatever the citation said? Because we're not going to like evaluate. It's like totally out of scope to evaluate in absolute terms whether a statement is true. You just evaluate based on what the citation says. And what you'll notice is number one, Wikicrow is way better than Wikipedia at citing its sources. So 16% of the statements in Wikipedia that we found like are just like not cited, right? I mean, this is should be consistent with your experience of reading Wikipedia, right? Whereas Wikicrow generates cited, basically generates citations like for everything it writes. Almost 97% of things it writes are cited. But Wikicrow is about 10x more less accurate. gets confused or makes stuff up. Doesn't exactly make stuff up. I made this point about saying it doesn't hallucinate. It is still true here that it doesn't hallucinate. What happens here is that it usually gets confused about what it's reading. So the most common thing is that it will be reading something about like the N-terminus of the structure of the protein, and the people will say, oh, it has like this protein has a unique fold at the C-terminus, and then at the N-terminus, there's this fold that is like broadly conserved among all members of this class. And then the Wikipro will write, at the C-terminus, there's a unique fold that is broadly conserved among all members of the class. And I'm like, no, that doesn't make sense, right? Like, no, it cannot be both unique and like widely, and very common. It doesn't make any sense. And it's just like something, it has like discarded, the model has somehow just like ignored the like N-terminus versus C-terminus distinction. We see that kind of thing a lot, where it's just like there are tokens that are important in biology that like the model probably hasn't like seen enough in its corpus or something. It will also do that with gene names. Like, rare genes, there's like piezo1 and piezo2. For example, it will like take a sentence about piezo2 and it'll just like stick into the piezo1, like, hey, this number on the end is definitely a typo, right? No. So that is like just really obvious, like super obvious ways to fix it. That accounts for basically all of these errors. I should caveat here, like in case you haven't noticed, this is 0.9% of statements in human-generated Wikipedia are incorrect out of 106 statements. So we found one. So we don't know how many errors there are in Wikipedia. I mean, we're like chat noise limited. I think certainly we know that Wikipro right now has more errors, but I can guarantee you basically like 3 or 6 months, like we'll do a better measurement of this, but 3 or 6 months is going to be on par with or better than the human-generated Wikipedia. And at the point at which machine-generated Wikipedia is like, or like Wikipro is both like more accurate than and also more comprehensive than human-generated Wikipedia, you kind of almost don't even need Wikipedia anymore. You just like you can imagine just like generating like, up-to-date accurate summaries of whatever you're looking for, like on the fly. I mean, I think we're like a few years away from really wanting to like replace Wikipedia, but like on the flip side, we're only a few years away from really wanting to replace Wikipedia, which is kind of insane. Okay, so just a word about future directions. So we are interested in a bunch of other things. I mean, I think that, again, just like I said before, the only thing that matters is whether you can evaluate your performance. And so what we're basically doing is we're trying to figure out, like with Wikipro, this evaluation that we're doing for Wikipro is like pretty cool. It is manual, but it's like pretty scalable. Like it's very easy for untrained humans to do. And so things that are automatically evaluable are definitely better. But basically what we're doing right now is we're just trying to figure out like how do you evaluate these, how do you evaluate all the various topics that we have to do. Everyone wants us to be coming up with like better like ideas. Like how do you how do you figure out like whether this idea is like good or not? Well, like I mean, we don't have any way to evaluate that right now. But like all of the big advances in this area, maybe slight exaggeration, but like not that much. Almost all the big advances in this area will come from like better ways to evaluate performance on these tasks, right? Because once you can evaluate performance in a really robust way, like the rest is kind of. I mean, I don't know. I don't want to curse myself by saying the rest is easy, but it's certainly, at least then it's like tractable, right? Before you have a way to evaluate, it's just intractable. And so, yeah. I guess happy to take any questions. Yeah, happy to take any questions. Yeah, thank you guys. Yep.
On the previous slide, we have a third column that shows that your wiki row for the pages that Wikipedia created, and then compare these two.
Sorry, you're asking, did we?
So, like that third column here, Wikicro results for the articles that Wikipedia has.
We did not do, yes. We did not do that. Actually, we did not generate articles. We did not generate articles for the, I'm sorry, if I understand what you're saying is, did you generate Wikipro articles for the articles that are that has human Wikipedia articles and then compare them.
That's right, so that you can compare those.
Yeah.
Crowdsourcing versus,
So we didn't, and the reason is, so first of all, I'm exaggerating slightly when I say that these 15,000 do not have articles. Most of them have stubs. I mean, like 2 sentence, 1 sentence, 2 sentence stubs. We count those in the list of ones that like don't have articles. Otherwise, like, there's a continuous range of human written Wikipedia articles. If you look at the human written Wikipedia article for KRAS, it's probably like 50 pages long, right? And you just like, you can't, whereas like for most genes that have Wikipedia articles, for most genes that have Wikipedia articles, like, you know, there'll be 1 or 2 paragraphs. And so, yeah, we didn't do it because it was not clear how to compare it. It didn't seem like it would be different also. It didn't seem like it would be different from, like, these statistics, I'm pretty sure would be the same. Or maybe not, because actually this is a good point. It might be more favorable because the genes that have Wikipedia articles are the more common genes and are therefore easier. So
if you can show the improvement over human beings that way.
that would be a better way. Yeah, that would be a better way to show the improvement. I think once we're there. Right. I mean, the point at which we can generate a KRAS article, an article for KRAS that, sorry, KRAS is like a really common like oncoprotein. It's involved in a huge number of cancers. The point at which we can show a like article for KRAS that like most experts will look at and agree is like better than the human written Wikipedia article for KRAS. I mean, that would be awesome. But like, you know, those articles are super high quality, right? And so, you know, we're a little bit away. Yeah, that's all. Great question. Yeah, so basically this is kind of similar. So this was paper QA, this is not WikiPrep, but like, you know, but basically we debated that question for a long time and we just came up with this taxonomy, which like what is generated when you get an answer from paper QA is a bunch of text that is like some fact or like some like statement, and then and then like some citations. And the hallucinations that we observed in the models in general would break into one of like 3 categories. One which we would call like a full hallucination, which basically just means this is all human evaluated, right? So it means that you read it and you're like, where does this come from? There's like nowhere this comes from. The statement is like not correct, right? This is just like, you know, when you don't have any other explanation, you call it a full hallucination. Then the other 2 forms are basically like citation, like the citation accuracy means that there's a statement and the statement is cited and that citation does not have any clear like that citation, it's like it's a citation that is not accurate, like the citation doesn't exist, right? Often you can actually kind of come to a conclusion about what paper it was referring to, so you can check. Like you're like there's a paper that's like close enough, and you can kind of figure it out. But that's and then you can like check whether the fact is in the paper. But so that's like citation accuracy. The context of relevance is that you get a correct citation to a paper that's just totally irrelevant. Those are the 3 citations, the 3 like kinds of hallucinations that we observe. I think it's like noteworthy that like the language models on their own, especially GPT-4 and Fog, like do provide accurate citations sometimes, and sometimes actually provide like accurate citations where like the information you're looking for is in the citation. It's just in the weights. I mean, that's cool. That's great. But like, if you guys haven't tried this, it's like worth trying. Like the kind of thing that will happen is like like when I try it on some of my papers, you get like the correct title, and then you get different authors who are like people who have worked on highly related topics, right? And we would call that like you know, citation accuracy, right? So yeah, other questions? Yeah.
Curious how the citation relevance
like in the next slide, what was that calculated for citation? Was 8%.
Oh, wait, in Wikicrow. So that is literally, I think it should say, in fact, it got corrected in the, it got corrected in the, on the blog post. It should say irrelevant or missing citation. So so basically we broke it down into like, we took Wikipedia articles, we like, randomly sampled, how we did this. I don't remember the details of how we did the sampling. I think we randomly sampled a Wikipedia article, and then we randomly sampled like a sentence in the Wikipedia article. And then a human went in, found that sentence, and looked at like the entire factual statement. Sometimes a factual statement consists of more than one sentence, right? But looked at like a cohesive factual statement. And asked, is there a citation to support that factual statement? And then this number is the fraction where either there is no citation, right? Sometimes it's labeled as like citation needed, and sometimes it's not. But there's no citation, or there's a citation, you go and you look at the citation, and like the fact that's referring to is just not there. Yeah, these are human written. So and again, I mean, like, people do this. If you've written papers, like you may have done this, where you're like, oh, I'm pretty sure the fact is like in this paper. I'm going to cite that paper. But like someone who actually goes back and looks and will be like, no, the fact is not in that paper, right? Like that happens all the time. But, yeah.
I mean, this might be an edge case, but how many times do you see that there's like a, for example, a correct statement, like a synthesis statement that does not cite all the relevant information?
In Wikipro or in human?
Well, I mean, Wikipedia does that all the time.
Wikipedia does all the time.
And Wikipro do better. Like, for example, like if you were to imagine writing a review, which all of these articles are, sometimes reviews would include synthesis of multiple different papers to, say that it's more than the sum of its parts. How many times is it able to correct it?
I know we don't have a good way to isolate sentences that are true syntheses. Like you can kind of see that it happens now and then. Okay. It definitely happens now. But we don't have a good way to isolate that, and so we haven't measured it.
Okay.
Yeah, Sam.
Yeah, I guess one question I had is you mentioned that evaluating RAG agents output is like having a systematic way to evaluate them is an important problem to work on. I guess like what steps are you guys taking towards that? Are you like shortening the time to characterize the things that the hypotheses that are being generated by these agents, or? Oh
no, I think that so in this case, like this number, we now have like a really nice protocol to just do it. It's humans, like I said, I mean, like I mean, we've definitely thought about whether you can have a model try to do it, but then you get into this like weird circle regime. It's just like not worth it. And so you can definitely, I think it takes a human. If I give you a statement, I have a nice interface. The interface is really important when you're getting humans to do things, right? You have a nice interface where you provide them a statement and you provide them like a paper, and you just, and like the text is there, the statement's there, and you just say, is this statement supported by this text? Right? And then they go through and they look. A human can do that in like 2 or 3 minutes. And so you're paying, that ends up being like $2 or something. I mean, like if you're paying a human $50 an hour, something like that. So or say $60 an hour is $1 a minute, right? So that's $2 or $3 per example. And so you like the first thing I'm doing is running a benchmark of just like, here are a bunch of statements, and or like here are a bunch of papers, and like can you come up with like can you come up with like correct statements? But basically, yeah, you can outsource the contract. It's really easy, right? Because it's like $2 or $3 per statement. Yeah, you can do this like this evaluation scale pretty well. So, yeah.
I guess this is more of a meta question, not really related to the work you've talked about so far, but especially hypothesis generation is one of those tasks where, like you said, evaluating is hard. So would you consider working on proxy tasks that would result in transfer improvements? improvements in
But
hypothesis.
how are you going to measure transfer improvements if you don't have a way to measure performance on the task? This is like my main objection, which is that like, it's fine. It's like great for research to say, you know, here, like science is hard and therefore we'll do Minecraft and it will just transfer. But like, will it transfer? Like, if you don't know how to measure performance in science, you don't know if it will transfer. So, I mean, I personally, I suspect that it will transfer to some extent, right? It would be weird if it did not transfer at all, but you don't know how much. And so the problem remains, like, these are independent problems. You can build a great Minecraft agent, but you still eventually need to be able to measure how well it does in science. Yeah.
How good is Wikipro at being able to link back to ground truth data? So what I really mean by that is, like, for instance, if you're going to say here's a list of Wikipro articles on all the proteins in the human proteome, how good is it actually at linking back to, say, NCBI and being like, okay, we're actually talking about this protein at this position on the human genome, and here's the sequence, here's a link for it.
Yes.
Good day.
We would be very interested in implementing that. So it doesn't do it, but well, I think, look, the kind of the dream is like for Wikicrow to then like go have it like see some statement, and then go and like kind of in a paper and be like, oh, is that statement actually true? And have it like look at the figures and like do some analysis, and yeah, we're not quite there yet, but definitely kind of on the roadmap. So yeah.
Yeah. What is your platform right now?
Yeah, great question. What's it for right now? So ultimately, what's the way that you evaluate hypotheses? I've made this point a few times, right? Like about like evaluating hypotheses. We don't know how to do it. I should have said we don't know how to do it in silico. The way that you evaluate hypotheses is you do some experiments. So basically what we're doing with the wet lab is we are building, like setting up very high throughput assays. So the wet lab takes a really long time, right? You can run 100,000, you can run paper QA 100,000 times in 2 days, and in the same amount of time, you will wait for whatever IDT to synthesize your oligos. And so the wet lab is really slow, but you can do things in high throughput. So what we're doing is we're setting up like super high throughput assays where you can get, hundreds of thousands, maybe millions, maybe tens of millions of data points on a 3-day, 4-day turnaround, something like that, so that you can start to generate hypotheses and you can evaluate whether you're doing a good job at generating hypotheses. Yeah, and more details on that are kind of like to be determined. But we're like starting to set up those assays and figure out what the right environment is. That was part one. I think you said you had 2 parts.
Yeah, and then part 2, what do you think is really differentiating between you and Perplexity Copilot right now as far as performance?
Oh,
Like, what stops Perplexity from training on Cognitive, for instance? Or like they do this to some degree.
what is differentiating with what's out
What
the
do you
group?
think is really driving the increase in performance? That previous figure that you showed?
Yeah, 2 things. We don't really know. So there are like definitely like some things about Wikipro that are just like optimized, or like paper QA that are optimized for the scientific literature. Like you can follow citation graphs and things like that. And we're like building some custom, some like custom embedding models that are specialized for the scientific literature. So we also have like access to a way larger full text corpus than they do, which like my guess is that right now that's actually the differentiator, is just like access to full text articles, where like there are probably some questions that like are not possible to answer without access to full text articles, which is where complexity falls down. That's my guess. But
Why don't they have access?
because they're paywall. And we have access through like through our own subscriptions and university affiliations and things like that. But like we would not be able to provide those, like we could not just like provide, it's not like they can go sign up for a subscription and then start offering it to the entire world. Like, we're using it for like internal research purposes. So it's easier. I do think like one point that I would like to make about these systems is that like, Perplexity is doing great because they're like pursuing a mass market. The market for these kinds of systems is like not large among scientists. There are like 100,000 maybe scientists in the world who will want a system like that. And so there are companies that are like trying to make similar systems and trying to sell them for like $20 a month. And so their revenue is like capped at $2 million a month if they have 100% market penetrance. And that's not enough to sustain a venture-backed company. And so this is part of the reason why we're doing a nonprofit setting. But Perplexity is doing a great, Perplexity, don't get me wrong, they're doing a great job. Yeah, you had your hand up before also.
Yeah, so curious in terms of like long-term vision of future mouse, is it to help scientists answer questions that they probably want to answer better, or maybe like long-term future help scientists with those?
I think that in like the long-term future, what we really want to be doing is building kind of like comprehensive mechanistic models for biology. And now what is a mechanistic model? I don't exactly know right now. And so I'll leave it there. But something about, if you had a mechanistic model, a comprehensive mechanistic model for the brain, like whatever that means, like what form it's in, what form it takes, I don't know, right? Then it would not even, the question would not even necessarily be anymore like helping scientists come up with like better questions, right? Like, because you would like have the oracle, and the oracle would be able to like give you answers to your questions in like some understandable like mechanistic form. It presumes that the knowledge is fully right. I mean, like, first of all, it's definitely not known. You have to conduct a lot of experiments to get there. And there's another question about whether it's like knowable, but certainly, I mean, that's kind of the like I kind of said at the beginning. I think that part of the issue is that if I said to you that you had infinite money and infinite resources in order to generate, even just like make it simple, like a full mechanistic model of E. coli, I don't think that you would necessarily be able to come up with all the experiments that you need to come up with, and like to be able to like sequence them in the way that would actually get you the data that you need to generate that model. That's like ultimately that's what I think that these things will help us do. Now I'm like really, I should also say I'm like I don't think that they will like replace human scientists. I think that humans who use AI, like human scientists who use AI will replace human scientists who don't. But ultimately, like what you said is probably like will also be true, which is that humans will always have questions and the AI will just like help us to answer. But yeah.
So like these RAG systems, like Pro or PaperQA, what's the underlying like model? Is it a pre-trained model, or do you call some API?
It's a mixture actually. So the different like pre-trained models are all good at different things. So like it was actually funny because you can like see as different checkpoints. If you have a good benchmarks and as different checkpoints become available, you can like see things change. And so in like September, it was a mixture of like GPT-4 and Claude, and then it just became GPT 3.5 Turbo and GPT-4. We've tried the open source. The open source models are like not good enough at all. They will get better. I mean, we would really like it if they get better. I'm sure they will, but yeah, at the moment it's mostly APIs that there's so much further. Yeah.
One thing I just thought about, like in terms of if we're imagining this as a tool to be used either by like biologists or anyone else who's trying to work towards some biological question, one problem that I frequently encounter is like I work in both the wet lab and dry lab, and a lot of my dry lab colleagues will come to me with genetics questions, and they will pull up a bunch of papers and citations for some gene of interest, and about half of them are low quality.
Yeah.
So, I'm imagining like, one powerful use of this, I don't know if it's on your radar, would be to detect such instances of like low quality data, falsified results, or otherwise like spurious observations that would, could
Absolutely.
people who are not in the
Well, this is like what I was saying, which is ultimately what you would like is you would like to find, you would like to have WikiPro like read a paper and then go and maybe like try to like see if it could look at the data, try to reproduce the analysis. One day, if it has access to a lab and if it's sufficiently high value, go and like do some experiments. I mean, I think at the point, like I made this point about like the $20 a month thing. The flip side of that is that like really high quality scientific summaries, are worth a lot of money, right? I mean, and so if this was like, hey, you have a month and you need to go and like answer this question, you really like have a really robust answer. And that means that you go, you see, summarize the literature, you figure out what are the key experiments that you need to reproduce. Or if you can reproduce those experiments, you'll have the answer. Then you go and run them. And at the end, you come back and you say, hey, look, this is the answer, and we're confident in this answer because we did this experiment. Because like this data that's published is really high quality, and we did this experiment, that experiment, and they reproduce, and therefore,
That's like half the legwork of doing a paper writing, like especially writing
Yeah.
reviews, that you're not just responsible for, showing you results, you also need to make sure your logic is square.
Yes, and it's also where the target, like validation comes from, right? In like drug discovery. It's like, some people have done some stuff in academia. Do we believe it? Is it robust? So, yeah.
You did this previously. I know the hypothesis generation and collaboration is the next big thing you guys are thinking about. It's very hard to do. But what are some internal ideas that you guys are talking about what hypothesis, which hypothesis is a good process for this?
I would say we don't have a bunch of very good ideas about in silico evaluations of hypotheses. We have some ideas. We don't have a bunch of very good ideas around in silico evaluations of hypotheses. If you want to know more, we can take it offline. I'm happy to tell you a little bit about more detail about what we're thinking. But I think that ultimately it will really boil down to like what lab evaluations, which means that you need to have some like really efficient like loop, right? You need to be learning a lot from every time you come with a bad hypothesis. You need to be learning a lot about what that is, about why the hypothesis is bad, right? Like this is part of what I was saying about how this is like an interesting playground for AGI, because like ultimately for this to work, like you can't do 100, you can't like test. You can do these poor assays that I'm talking about, where you get to test effectively 100,000 hypotheses, but you can't test 100,000 different genomics. It's not possible, right? So, yeah.
Everyone else.
No, of course. Yes, you have a question, I guess.
So like how does it like produce like configured results, like if the results produce?
Sorry, I can't hear you.
So I was wondering like if like here's how I turn results, like configured results, is your mechanisms like weight differently or like some way to turn it?
We have not, we not yet. And honestly, we haven't exactly seen it. It's relatively uncommon that two papers will directly contradict each other. I'm sure it's more common that you have like 5 papers where if you actually put the results all together, you'll get some like, it'll be overspecified. You'll get some like, it'll be mutually contradictory, but we don't, it's hard to find. We haven't seen it. Yeah.
I guess I just wanted to follow on the hypothesis generation thing. Have you thought about going in the reverse direction where you provide some, I don't know, some data, some results, a description of these things and be like, what could explain that?
Yes, actually, we have one person who's working on it. I'm not up to date on the results, but if you're interested in that, I can put you in touch. Like, I think it is.
That's the way that we would write questions to evaluate students who are supposed to be learning experimental design.
Yes. Yeah, actually, I do know that there's one person who's been playing around with it, but I don't know. Yeah. Anyone else?
All right, let's give a round of applause to kind of.
I do that. Oh, and I should say, so we have like, we're looking for, so first of all, we're hiring. So if people are excited, about this kind of stuff and want to come work with us, like, let me know. We are also looking for interns, and if you we've been like kind of putting our internship program together slowly. And so if you previously like, we had an opening, if you applied to it, or if you emailed us before and you didn't hear back, just like email me again. Because that's been like coming together kind of slowly, but we're getting it more together now. And so, and just to emphasize again, we both do research and also pay money. So, yeah. Okay. Thanks, guys.